Good evening. My name is Diego, one of Bergamo Scienza volunteers. Before starting, we would like to kindly ask you to remember that you can connect to the Bergamo Scienza site.it, and clicking on the live button, you can follow the live event on Facebook and Twitter and YouTube. You can send questions to the speakers and invite your friends and followers to follow the event online. Questions will then be selected by us, the volunteers, and we'll be posing them during the question and answer session. For simultaneous translation, you can scan the QR code that you can find on the doors of this hall or projected behind my shoulders. The QR code will address you to the Converso web app, where you can select the translation language from the scroll-down menu. To listen to the translation, you need a pair of earphones. You should kindly use your own. If you don't have them, you can ask our volunteers. You're all invited to follow our Instagram profile, where we'll be publishing our events. You're kindly asked to mute your devices and attend the Bergamo Scienza events. Thank you very much for your collaboration, and enjoy the evening. Good evening to you all. Tonight we'll be speaking about cancer, and we are lucky and honored enough to deal about cancer with Professor Laurie Glimcher. She has a degree at Harvard University, and she was the dean of Cornell School of Medicine, first woman. And then she went back to Boston, and she's now the CEO of the Dana-Farber Cancer Institute, one of the most important cancer institutes in the world. Besides these prestigious jobs, she's also a researcher, and she's been dealing with innovative aspects related with cancer treatments, in particular immunotherapy, the regulating mechanisms of our immune system that, when stimulated by means of mechanisms that have been recently discovered, can be used to use our immune defenses to fight cancer. And these immunotherapies have been revolutionary because the prognosis has been changed thanks to them. This is what she has been dealing with as a researcher, but Professor Glimcher is also an advocate of access to treatment policies. Yesterday evening, we discussed these issues. These therapies are very expensive, and the mediation between the pharma industry and the possibility to grant universal access to everybody is definitely a huge problem, with some clear differences between healthcare in Europe and in the United States. I do not want to steal any more of your time. I leave the floor to Professor Laurie Glimcher, and she will explain the end of the beginning, this new frontier in cancer research. Hopefully, we manage to reduce cancer death rate by 50%. The objective was set by President Biden, and this within 2040. The floor is yours, Professor Glimcher.
and prego. It is a pleasure to be here. This first slide simply tells you that I do some things outside of running a cancer hospital. I also consult for some of these companies as well. So tonight, we're going to talk about cancer and curing cancer. And I call this the end of the beginning because the last 2 decades have brought us very far along in treating at least some cancers. Now, let's set the stage. So this is really the most important time for cancer treatment, cancer research, cancer care. The high incidence of cancer in the United States, 1.7 million cases annually in the US. So cancer actually is the leading cause of death in patients aged 45 to 64, and people over the age of 65 still die most frequently for cardiovascular, heart attacks, strokes, but within a few years, they too, their major mortality will also be cancer. So let's think about how this affects everybody. 1 out of every 2 men and 1 out of every 3 women will develop cancer during their lifetime. So this is a very, very common disease. What particularly strikes me now is the growing incidence of cancer in young people. So the death rate and survival rate for adults has gone down. That's good. Less mortality for a number of reasons which we can discuss. But in the last few years, we have seen healthy young men and women who present in their 30s, maybe in their 40s, who present to Dana-Farber with metastatic colorectal cancer. And we haven't yet figured out why that is. That is also true of young women in their 40s who are non-smokers and who never smoked, also have an increase in the incidence of lung cancer. So let's just look at the whole picture. Over the last 2 decades, there have been 2 revolutions. One is precision targeting of a genetic mutation, and the second is activating your own immune system. So we've done pretty well in treating those, and you can see this here in the gray. We can treat these cancers quite well. Most individuals will survive with melanoma now, with thyroid cancer, with prostate cancer, and breast cancer. But there are a number of cancers, some of them very common, that we really haven't made any progress in. And that includes pancreatic cancer, which is almost uniformly lethal, brain cancer called glioblastoma, which is almost always lethal, and we still have many patients with lung cancer, ovarian cancer, that we really do not know how to treat. If we look at Italy, here are some statistics for cancer in Italy of a total population of over $60 million. not dollars, 60 million people, wish we had $60 million. The number of new cases are over 400,000, and the number of deaths is almost half of all those that develop cancer. If you look over 5 years, the existing cases are high. They are 1.2 million patients who have cancer. Now, the cancers are pretty similar to what we see in the United States. Breast cancer, colorectal cancer, lung cancer, prostate cancer are the most common. If we look at men versus women, in men, the most common cancer is prostate cancer, which is good because we usually can either cure prostate cancer or we have enough new drugs that can keep it in abeyance. So many, many people over the age of 70 have prostate cancer, but they don't die of prostate cancer. It just kind of sits there. Lung and colorectal cancer are also very prevalent in men. In women, to no surprise, breast cancer is the most common cancer. And now we can do pretty well with breast cancer thanks to a number of new treatments that have been discovered. Women also have high levels of colorectal cancer and lung cancer. But the good news is that, and I'm just going to go back for 1 second, is that the survival rate of individuals with cancer in Italy are better than in the United States. And that may be due in part to superior medical care, or it could be due to the fact that you have the best pizza in the world. Now, the good news is that we now have many, many more cancer drugs than we used to have. So in the last decade, about 100 new drugs for cancer have been approved by the FDA. For example, in lung cancer alone, in 1 month, there were 7 approvals of new treatments, and we're going to talk about what those treatments are. And that includes as well new drugs and biologics. So we're on a good path. For multiple myeloma, which is a fairly common disease of the blood, there have been 26 new treatments over the last decade. Most of them, I must say, occurred at Dana-Farber Cancer Institute. Patients with multiple myeloma 20 years ago had a lifespan of about 2 years. Now, some of those patients can be cured with the current therapeutics, and those who aren't cured have an average lifespan of 10 years. So a lot of progress in certain of our cancers. Now, I want to talk about cancer treatment, and I'm going to cover, at least briefly, each of these 4 important new research that have led to the development of these cancers. One of them is cancer genomics. A second one is called epigenetics, which allows genes that were quiet to come alive and combat cancer. And then my favorite one as an immunologist, which is immunotherapy. We'll talk about that. And finally, we'll talk about something that is very new and is not yet well established, and that is figuring out how to detect cancer very, very early on in stage 1, because only 25% of our patients now present to us with stage 1 cancer, and we can cure those patients. But the other 75% come to us when the tumor has already spread to other places in the body, and it is much harder to treat those patients. We can treat many of them and even cure some of them. But the future for me and for all cancer patients is let's detect that first cancer cell right away, because then we don't have to do anything else about cancer if we can detect it extremely early, and I'll talk a little bit about that. So let's start first with what we call targeted therapeutics in cancer. So if you think about how we attack each disease, let's say it's heart attacks, strokes, cancer, infections, we tend to focus on those diseases that are most common. And the most common disease is cardiac and stroke. And so researchers immediately dived into that 50 years ago in an intense way. And now, look what we do. We have reduced the number of heart attacks. We have lowered our cholesterol and lipid levels. We have figured out ways to immediately treat strokes. So that was in, let's say, the 70s and 80s. Cancer back in the 90s was at a dead spot. We really hadn't accomplished anything for cancer despite the cry for treatments in the Nixon administration. Nothing really happened. So we were really stuck in a hole and nothing happened. But in the 90s, all of a sudden, the field opened up. And the first, what I'll call revolution, is to talk about targeted drugs. So cancer is a disease of your genome. DNA, half of which you inherit from your mother and half from your father, is what we call the genetic code, 4 letters, all right? Now, we have 6 billion of these letters, and they encode 20,000 genes. So humans have 20,000 genes, half from mother, half from the father. And what goes wrong? And this was the realization that genetic mutations in DNA, even one single genetic mutation, can lead to a cancer. So just looking at this, how does that happen? So a mutation can happen because you have a misspelling. Should be dancer, but it's cancer. Or the gene amplifies several times, and you get 101 Dalmatians, which I think none of us want. Or translocation, when you take a piece of DNA and move it to another chromosome where it misbehaves and results in cancer. So let's take a look at this. Here's DNA, the helix. So mutations in even one piece of DNA will change the proteins that emerge from it. As DNA gets transcribed into RNA, RNA gets translated into proteins. So here we have an active protein and an inactive protein. And the problem is some cancer-related mutations have changed the DNA code such that they keep a protein in an active role instead of toggling back from active to non-active. And having a protein that is continuously active and always churning away can lead to cancer, because these mutated proteins are sending signals to the normal cells that encourage them to grow and divide and become malignant. That's what cancer is. So, how do we stop that from happening? Thanks to the human genome, where a number of brilliant scientists with much help from many academic medical centers, including Dana-Farber, sequenced every single one of those 20,000 genes. And we sequenced them not just in normal people, but in cancer people. So we did hundreds of thousands of sequencing of the genome and of the tumor in patients with cancer. And that allowed us to identify what is the genetic mutation that this patient has that is driving their cancer. And then we say, okay, what can we do to develop a drug that will disable that mutation? Because that mutation is driving the proliferation of tumor cells. So if we can find the right treatment to disable that mutation, I'll show you what happens. So this growth protein that arises from the mutation, is pushing the cells to multiply rapidly. But if you have a chemical, a small molecule chemical that prevents that, the tumor shrinks and disappears. And we know that this works, and this is one example of patients who have genomic mutations in lung cancer. And about 30% of patients with lung cancer have the same mutation, and it's in a gene called EGFR. So a drug was developed called osimertinib, and this was actually developed at Dana-Farber in collaboration with Novartis Pharmaceuticals, developed this drug, and they looked at its effect. So this is the probability of progression-free survival. Patients who did not have that mutation, these, you can see, did not survive well. This is the progression. Patients who had the mutation and were treated with osimertinib, they weren't necessarily cured. Some of them were cured, but they had a longer lifespan. And here's an example of a man who had melanoma, which is a skin cancer, which is fairly common, and it's usually due to exposure, too much exposure to the sun. Having sequenced all of these patients, it turned out that 50% of them had a genetic mutation in a gene called BRAF. So a drug was generated that attacks the BRAF gene and disables it. And this looks what happens. So all of this, all of the red is the cancer, which is not only in the lung, but has metastasized to the bladder. And you look at a few months later, and the tumor is gone. Even though it is metastatic, it has spread widely. This patient would have died within the next 6 months, and instead, he is cancer-free. But here's the problem. Here's the problem, which really is a focus, an intense focus amongst the cancer researchers to figure out how to fix this. So here's a gentleman who has multiple myeloma. I'm sorry, who has melanoma. It's a skin cancer, as you can see. Before he was treated, spent 15 weeks taking the BRAF inhibitor, and look at him. It's gone. It's gone. And we said, oh, that's fantastic. But you just wait another couple months, and the melanoma is back. And why does that happen? It happens because another mutation has arisen. So the initial mutation was BRAF. They fixed that one with a drug, but it came back. Now here's another example of a man who had lung cancer. As you can see there in the orange. And he was given a drug that targets the EGFR receptor. The title isn't important. The point is that 30% of patients with lung cancer have this specific mutation, which we discovered by sequencing all the tumors of all the patients that came to a number of different cancer centers. So a drug was produced that disables this gene, turns it off. So here are all the people who have lung cancer. When they go on chemotherapy alone, most of them die. Only a few percent are alive 5 years later. It's about a 6 to 12 month survival. And my mother had lung cancer, and this was 20 years ago, before we had these targeted treatments. And she died, like most, at about 16 months. But if you put them on the EGFR inhibitor, now look at this. You have, and this is over almost 3 years, most of these people are still alive and well. About 20% of them are alive at 5 years. So you have given that group of lung cancer patients 5 years. So we haven't cured them, but we've given them 5 more years of life. So that's good. That is certainly better than before, but 80% of them are not alive, and their cancer has returned. So why does that happen, and what can we do about it? The cancer has become resistant to this drug treatment. And why is that? Because it has now received another genetic mutation. So genetic mutation number 1, they're now resistant to that drug, but within time, the tumor is smarter than we are, and it just comes up with another genetic resistance. Because cancer cells actually act and undergo Darwinian evolution. You see them once, they have this mutation, and then you see them 6 months later, and they have now a second mutation. So, you know, it's not surprising. There are 3 billion DNA base pairs, and they have to be copied every time a cell divides. And there's a small error rate. It's small, but it's 1 in every 100 million base pairs. So there is a constant rate of new mutations for all of us, but they're not uniformly distributed. There are hotspots. So what happens when that happens, which it's going to do? So here's the man who has the lung cancer. He was treated with the EGFR mutation. The lung cancer went away. He then evolved a second mutation, and the tumor came back. But then we were able to generate a second drug treatment that pinpointed the new genetic mutation. And so now he, again, is cancer-free. This is why it is crucial for cancer hospitals to sequence the tumor DNA. We need to know what the mutation is. Is there a drug out there that can disable that genetic mutation and prevent it from initiating cancer? And if not, can we make another drug that will inhibit the second drug? Now, I have to point out that this is not covered by insurance, and you might ask why? Because sequencing these patients allows us to treat at least some of them and to prolong their lives and perhaps even cure it. But healthcare system in the United States will not provide the funding to do this. So we actually raise philanthropy so that we can sequence every patient that comes through the door. So we've made progress. This is really revolutionary that one has discovered that it is genetic mutations that drive most cancers. Not all cancers, but most cancers. We can make drugs that can either cure the patient or give that patient more years to live. But eventually, almost always, the tumor pulls ahead by making another genetic mutation. So when you think of that, if patient number 1 has the EGFR mutation and then has a BRAF mutation, how many others will have the same one? And so that requires sequencing a number of people to find out what usually happens if you relapse from EGFR treatment. Well, it turns out that a lot of people have the same second mutation. So now we think, why don't we treat them at the beginning with both drugs? And this really was made clear by the HIV epidemic. So a 20-year-old now who becomes infected with HIV has a normal lifespan. And that is because the virus is attacked by several treatments, several drugs all at once. And so the virus does not mutate. So I think that's part of the future of a revolution So we need combination therapy, and we can get combination therapy, but not just from treating patients with 2 or 3 different drugs simultaneously. That is one way to go. Another way to go is called epigenetics, and I'm not going to spend a lot of time on this, but epigenetics controls the expression of genes. So every one of your cells has, I forget, 2 miles worth of DNA, and it's all compacted into a ball. And if you look at all the cells in your body, you have, I forget how many miles. Basically, you can go to the moon and back 40 times. That's how much DNA is in your body and is in each cell. But we have to control those genes, and that's what epigenetics does. And I'll give you one example. Monozygotic identical twins have inherited the same DNA from their mother and their father. They should be identical, but you know, when you look at identical twins, they don't always look exactly alike. Their personalities may be a little different. One of them may get a disease that the other one doesn't get, but they have the same genes. So how do you explain that? You explain that through epigenetics, because epigenetics activates or inhibits your genes, your proteins. And that happens while you are alive. And it's probably the reason why identical twins may have differences in susceptibility to their personality, to the disease that you have. You may have one twin that develops ovarian cancer and the other twin doesn't. So you get changes over time. Now this is in its very early stages, but because you want to turn your 20,000 genes off or on, that's called gene expression. And that's what epigenetics does. So this is a very hot field right now, and I think new drugs will be made based on this. And here's an example. Here's a child. He was a 9-year-old who had pain in his hip, and eventually it was so painful he couldn't go to school, and they gave him the first ever epigenetic drug that changes the expression of a number of different genes. The child got better, went back to school, pain-free. He's still going to school, and as far as we know, he's still alive. So I think enormous possibilities. And here's an adult treated with an inhibitor. She has leukemia. This is an inhibitor that was developed by one of our faculty members, Scott Armstrong, together with a company called Epizyme. And she had tremendous leukemia spreading through her skin. She was treated for 4 cycles, and they've disappeared and have not reappeared. So two things we want to think about: the next generation of genetic mutations that we develop drugs for, and secondly, gene expression. And now I want to come to what I know best, which is the immune system and immunotherapy. And here's a picture from Science magazine when the first discovery was made. Here is your cancer cell, and is surrounded by a specific immune cell called the T lymphocyte or T cell. the T cell is the brains of the immune system. There are a lot of different types of immune cells, but the T cell is the one that should recognize the tumor, see that it is foreign, just as it sees a bacteria or a virus that is foreign, and should kill it. And I want to go back to a little of the history, because over a century ago, a surgeon named Dr. William Coley, he was in New York City, was treating some patients who had a disease, a cancer called sarcoma. And he removed the sarcoma and then waited to see if they would recur. And most of them recurred, but he noticed that those patients who developed a bacterial infection in the wound, sometimes the tumor never came back. And so he said to himself, there must be some system in our body that recognizes foreign proteins, whether they're on bacteria, viruses, or cancer cells. And you've activated that immune system to kill off not only the infection, but also the cancer. And this was a long time ago, and frankly, people made fun of him. They didn't believe it. He bottled it up in a lysate called Coley's toxin, and, you know, asked people to buy it and use it, and there's a lot of skepticism. It worked a little bit, but then at that point, chemotherapy and radiation came into view, and this was abandoned. And for 100 years, scientists have been trying to figure out how to wake up the immune system. So you would expect that your immune cells, particularly your T cells, would recognize a tumor because it is covered with proteins that are foreign. And the amazing thing about the immune system is that it can recognize what is self and what is non-self. Non-self meaning a virus, a bacteria, and it will kill off that virally infected cell or that bacterium. So why doesn't that happen in cancer? Because cancer cells have lots of foreign proteins on them. So ordinarily, an immune system could do this. It sees a cancer cell, but the cancer cell gives the message, don't attack me, I'm you. And when that happens, the cancer grows. Now, of course, there are diseases, autoimmune diseases, where your T cells fail to recognize your own tissue. And that can lead to autoimmune diseases like multiple sclerosis, rheumatoid arthritis, and so forth, and systemic lupus. So began to look at this special cell, this T cell, that has the capacity to kill cancer cells. And when you looked at the tumors of many people, it was clear that if you had T cells that were inside the tumor, surrounded the tumor, patients lived longer. If you didn't have any T cells around the tumor, patients did worse. Immunology is a relatively complicated field, but basically there are two kinds of cells One is from the innate immune system and one is from the adaptive immune system. The innate system comes to the defense very quickly. The adaptive immune cells come in later. We needed to know, what does the T cell have to have in order to be active and kill off that tumor cell, that virus, that bacterial? And we were very fortunate in my own laboratory to identify that master regulator, which is a protein which we named Tbet. And it is a master regulator that makes that T cell ready to kill foreign cells, including cancers. And we noted that in preclinical animal models, if you take away that protein, which we called Tbet, those animals developed colorectal cancer. And we found a patient who genetically lacked Tbet, and that individual had compromised both adaptive and innate immunity. So when I think of the tumor and its surrounding environment, I think of the tumor as being a castle, and all around the castle is a moat. And that moat is filled with many different kinds of immune cells, but they are inactive because the tumor is greedy. The tumor gobbles up all the oxygen and all the nutrients in its environment, and it secretes toxins that make the immune cells ineffective. So the goal is to change that. And the two individuals who figured out a way to activate T cells, even when the tumor microenvironment is very exhausted, are these two, Jim Allison from Berkeley and Gordon Freeman, who is at Dana-Farber Cancer Institute. And what they did was to say, let's not just directly activate the T cell, because that's too powerful. That was tried in a trial in the UK, and patients died because they were overwhelmingly far too dangerous and active. So what Jim and Gordon did was to say, let's find inhibitory receptors on those T cells that make them exhausted and block them. So it's inhibiting the inhibitor. And that's where these wonderful compounds, they're biologic antibodies against these two inhibitory receptors that blocks them, revolutionized immunotherapy. This is really the first immunotherapy. And it was pretty amazing. So you're inhibiting the inhibitors. And here's an example. This is a patient who has lung cancer, which you can see is right over here, who received an immunotherapy trial with antibodies that inhibited the inhibitory receptors, both of them, Opdivo and Yervoy, and this patient has near complete resolution. Now, how long this patient will live depends. Some patients live for a long time, other patients don't. So, you know, this changed the world. This was a Nobel Prize, the idea of blocking a blocker. But only 25% of patients respond to these immunotherapeutics. And only 10 to 12 different cancers can respond. So glioblastoma doesn't respond to immunotherapy. Pancreatic cancer does not respond to this. So we clearly need to come up with other ways to activate the immune system. This is a great start. It is transformative, but it's not enough because the majority of patients do not respond to this. And when I think of cancer today and tomorrow, we've achieved good checkpoint blockade, but we need to identify more of those inhibitory receptors and make drugs, antibodies to block them. We also need to change this exhausted, hostile moat that sits around the castle and prevents the immune system to do its job to kill the tumor. There is also what you probably all are aware of as adoptive T-cell therapy, or CAR T-cells, cancer vaccines, early detection, and we'll talk about, let me just look here. Let's just talk about them fairly briefly. So here is what I call the moat. All the inhibitory immune cells that surround the tumor and are immunosuppressed. They can't help, they can't kill the tumor. Here's the tumor, and it's surrounded by innate immune cells like dendritic cells and macrophages, and by T cells. But these T cells sometimes can't even get into the tumor because there are so many other proteins that are made, like cytokines and so on, that make this a really hostile neighborhood. And you can see that again here. There's the tumor in the middle, surrounded by all these cells, but the T cells are exhausted or they can't get in. The dendritic cells, which are critical to activate the T cells, can't do it. And there are all these small molecules called cytokines that are also immunosuppressive. So there's been a lot of research into understanding how we can reprogram this hostile neighborhood and turn it into a neighborhood that actually can activate your T cells and kill the tumor. And one of those pathways is something that we worked on and identified, which is a kinase that, interestingly enough, when you delete this particular kinase, two things happens. The tumors die, and the immune system becomes activated, what I call a double whammy. And so this is a project which one can use to come up with drugs that block these two proteins. Now, what about adoptive T-cell therapy? Well, this is the living drug, right? You take a person, and this is only for hematologic malignancies like lymphoma, leukemia, multiple myeloma. It doesn't work in solid tumors. You take blood from a patient who is basically, this is the only thing left to do. You've treated with other drugs, you've even done a stem cell transplant, but the patient still has leukemia. So you take blood from that adult or child, and you manipulate it by putting certain targets in it. And then you expand those T cells and you put them back into the patient, where they will now force that T cell to come right to the tumor and kill the tumor. About 10,000 people have been treated this way. It is extremely expensive. It is really not accessible to that many people. The drug itself, the living drug, costs $300,000 to $400,000. So it's a boutique. It's still a boutique, but it has saved the lives of children with leukemia and adults with leukemia, lymphoma, and now multiple myeloma. There's a lot left to do to make this something that is accessible to all and much easier to manufacture. This is a GMP facility that we have at Dana-Farber that can make these CAR T cells. And then there's cancer vaccines. Now, when I'm talking about cancer vaccines, I'm not talking about preventive vaccines like the ones you get, measles, mumps, rubella, tetanus, whooping cough, that we all get when we're young, and then we remain protected from getting those diseases. These are therapeutic cancer vaccines. It's a very new field. But there is some hope. This MD, PhD, Dr. Kathy Wu, decided to make a vaccine that she could use in 8 patients who were virtually at death's door. They had widely spread melanoma. They had received everything possible, and they were going to die. So she took their tumor and looked for all the foreign proteins that were on each patient's tumor, because it differed from patient to patient. And then she added what's called an adjuvant, which activates the immune system. And she injected each patient with their own special tumor proteins, and also did it together with the checkpoint blockers. All 8 patients are alive 6 years later. They were on death's door. They are still alive. So Dr. Wu is now making cancer vaccines for pancreatic cancer, for brain cancer, for kidney cancer, for ovarian cancer. These are now in clinical trials. So I think this is the first example of a cancer vaccine that actually works. May not work on everybody, but the data so far suggest that it's very promising. Finally, let's talk, before I want to come to this slide, I want to make the point that it's great to have a therapeutic vaccine. That's great. But wouldn't it be amazing for our children and our grandchildren and our great-grandchildren to be able to have preventive vaccines against cancer, so that just as you're getting your measles, mumps, and rubella vaccine, you can also get vaccines that prevent you from ever getting cancers. That is the Holy Grail. And that brings me to another area of cancer therapy that is very new, but is also exceedingly promising if we can figure out how to do it. And that is preventing cancer to begin with. This is aside from vaccines, which are going to take quite a long time to arrive. And as I said earlier, if you present very early, stage 1 cancer, you have one lump on the breast, it hasn't spread to the lymph nodes, it hasn't spread everywhere else, or you get an x-ray of your lung for other reasons and the radiologist says, oh, you've got a little nodule here, and you get it taken out. But that happens to only about 25% of our patients. What if we could detect cancer really early? And that is what we and others are looking for. Now, some people have argued that we should be doing what's called a liquid biopsy. Let me go to the next slide. Liquid biopsies. Recently, it's been shown that in patients who are known to have, let's say, lung cancer or pancreatic cancer, whatever, you can just take their blood out. And cancer cells do die, and when they die, they extrude the DNA, including the genetic mutation. So if you just take a blood sample from a patient with stage 2 or 3, and you look for genetic mutations in those cells, then you know that patient has cancer. Very exciting. We have to get it to the stage where we can detect not stage 2 or stage 3 cancer, but the earliest forms of cancer. And a number of companies and academic medical centers are working hard on liquid biopsy so that you can take DNA from a few blood cells and see whether there's a genetic mutation. I think that's really the next horizon. Now, you can't do it on everybody. You can't go to your primary care doc and say, I want to get a liquid biopsy, because the false positive rate is too high. There'll be too many people who are told that they might have cancer, and then they have to go through a full workup with CT scans and MR scans and this and that, and suffer for the thought that they might have cancer when many of them will not. It's not affordable. It is simply for any healthcare system. So we're focusing at Dana-Farber on people who have very high risk of having cancer. So 10% of cancers actually are inherited. And you heard yesterday from Greg that for Alzheimer's disease, 5 to 10% of patients have inherited genes. The same is true for cancer. So those are high-risk people. If you see an individual who comes in with a specific mutation that we know leads to colorectal cancer, and then that person has that mutation, we get all the family to come in, because we want to know every member of the family, uncles, aunts, brothers, sisters, cousins, do they have that same inherited genetic mutation? Because if they do, we're going to follow them very, very carefully. Those patients have an 80% chance of having colorectal cancer or pancreatic cancer. So we say to them, you know, the age for getting a colonoscopy is 50. It's now gone down to 45 because we're seeing more colorectal cancer in younger people. So you're going to have a colonoscopy every 6 months. It's not the best experience of your life, but it's far better than being told that you have metastatic colorectal cancer or pancreatic cancer. So to me, this is really one of the things that's most important. The other people who are exposed, who have high risk, first of all, those of us we're all getting older, but if you're over the age of 65, the chance of you getting cancer increases. If you've already had one cancer, you're highly likely to get a second cancer. If you have exposure to carcinogens like tobacco, and now obesity, because obesity leads to inflammation, and it is going, the contribution of tobacco to cancers is unbelievable. 30% of all cancers are because of smoking. Obesity is going to supplant smoking as the biggest risk for cancer. So that's really what I wanted to focus on today. Here is really the bottom line. We can show that drugs that target a genetic mutation can lead to a temporary remission. We need to figure out how epigenetics that control the expression of many genes, turns them on, turns them off, have to combine those. We need to develop more immunotherapeutics, whether it is vaccines, CAR T cells, checkpoint blockade, and then we have to combine those treatments, multiple treatments at the same time, to hit the tumor and to activate the immune system. And finally, let's detect cancer early, so at a time when it can be cured. Now, before I complete this talk, I noticed last night how many young female women were here and who asked really the best questions. They were remarkable. And as a female scientist who runs a cancer center, I gotta tell you, it was tough. It was tough to, particularly 30 or 40 years ago, when women, very few women were scientists or doctors. And there was a lot of prejudice against all of us. So I just wanted to say to the young women here today that actually you can do it all. It won't be easy, but it can be done. And I love this quote from our Congresswoman from Colorado, Patricia Schrader. A colleague of hers asked, how can you be in the US House and still be the mother of 2 young children at the same time? And she replied, I have a brain and a uterus, and I use them both. And it can work. Those are my 3 children early on. They seem to have managed to have a mother who wasn't always home. That's what they're doing now. And they have produced these 5 grandchildren. So don't give up. It can work out, it will work out, and it's getting much better than when I was a young scientist myself. So thank you very, very much. Mille études.
Bene.
Well then, let's start with the questions right away. So let's start with the questions right away. I see that, can you hear the translation? Hello? Testing, testing. Can you hear the translation? Can you hear the translation? Since we're facing this monster, cancer, is there a role for surgery? To treat cancer, is there a role for surgery? He's repeating the question. When facing such a monster, cancer, is there a space, a role for surgery? Because you didn't mention surgery at all tonight.
The question was whether there is a role for surgery here, and the answer is absolutely yes, particularly in stage 1 cancer. So if you come to the hospital and you have a nodule in your breast and you biopsy it and it is cancer, you will have that, either have the entire breast removed, or if the lymph nodes are negative, you'll just have what's called a lumpectomy, which is just taking out the cancer. So many patients who present with cancer do undergo surgery. If the tumor has spread to multiple places, then most of the time surgery is not performed and the patient is put on a variety of drug treatments because the tumor is already spread and you just can't get all of it out.
Buonasera e grazie.
Thank you for your presentation. What's the role for mathematical models in trials on new cancer drugs?
I'm not sure I got this right. Are you asking about clinical trials?
The mathematical models.
What is he asking for?
He's asking if the role, potential role of mathematical models in clinical trials.
A very good question about mathematical models in cancer. And they're extremely important. And I think, you know, there's so much data to collect about every patient with cancer so that we can uncover new ways to treat them. And that means we need to know about their medical record, and we need AI and natural language processing to be able to put the medical record in terms of a few algorithms. We need to take our x-rays and our pathology and look at them with artificial intelligence. We need to look at patients' metabolites. We need to look at their genome and their immunoprofile sequence, because these are going to be big baskets that will necessitate mathematics, artificial intelligence, natural language processing in order to gather enough data about enough patients that we can figure out eventually, at some time in the future, when a patient first comes in and you do all those tests on him or her, that we will know from the thousands of patients that we've collected data from exactly what drug you should be put on, what drug might work for you, but you can't have it because it'll be too toxic, or the drug is not going to work for you. And I think there's an explosion in that. We do 1,100 clinical trials at Dana-Farber a year. And many of those trials, over a third of them, are phase 1 or phase 2 early trials because they are innovative and investigated initiative.
You explained that oftentimes when there is a mutation and you give a certain drug, then often a second mutation comes about and cancer recurs. But why is there a second mutation? Could you please explain? Is it because cells then proliferate quickly, thereby making mutations more likely? Or is it because in the patient's body there is something making that patient more susceptible to genetic mutations?
She's asking why after the cancer cell mutates once, then you treat it and then it mutates again a second time. Is it because is that genetically determined or because cancer cell grows fast?
Yeah. So the question is, why do we have second mutations? And the answer to that, which is a great question, is that the tumor is smarter than we are. They go through so many cycles, they proliferate so fast that there are bound to be additional mutations. And the body selects that mutation, the second mutation that allows it to grow. So that's a clone of a cell. It's got a second mutation which allows it to circumvent treatment. And that little clone will go bigger and bigger and bigger. So it's really, you know, the faster growing mutation will dominate any other mutations. Because cells, even normal cells, divide all the time. And DNA gets changed. Mostly when DNA gets changed, it doesn't hurt you, it's not important. But for the cell that now can grow out of control, that's what happens. It takes over because that mutation allows it to grow quickly.
Buonasera.
Good afternoon. I have a question about drug combinations. Are they used for immune treatment or gene treatment? Are these drug combinations effective? If so, for which tumors? And then in case of these multiple gene mutations, I wanted to know whether multiple drug combinations are given to try and prevent proliferation from occurring.
He's asking about...
In case of mutation, besides gene therapy, if you can also combine with immunotherapy.
Oh, yes, yes. So the question is, can we combine the approach of developing new drugs for new genetic mutations with immunotherapy? And that is exactly what we're trying to do. We need to hit that tumor cell in 4 or 5 different ways, just as the HIV virus was attacked by several different drugs. So yes, it's a combination of all of what I've told you, whether it's early detection, CAR T cells, cancer vaccines, immunotherapy, and targeted drugs for genetic mutations and epigenetics. So the goal is to combine them together in the hopes that that will prevent them from developing new genetic mutations.
Un'altra domanda lì.
Thank you, Professor, for your talk. Very interesting. I wanted to ask actually two questions. One on commercial screening, commercial DNA sequencing, like Illumina's product. Can they detect phase 2 and 3 cancer? So this type of screening or DNA sequencing technique. If so, do you think that the overscreening could be an issue like in the coming years, as we have clearly a drop in price for this kind of system? And then in the FDA timeline that you mentioned, the long timeline that you have for FDA approval, do you see simulation, so like digital twin of the body, being applied in your laboratory to do simulations basically of your system to prove that it can be used without any drawbacks? Thanks.
Okay, that's at least 3 questions. I hope I can remember all of them. Illumina, the company, does a lot of sequencing for many academic medical centers. So we either use them or we buy their equipment to do the sequencing, the genetic sequencing. Illumina recently purchased Grail, and Grail is a company that is focused on early cancer detection by liquid biopsies. I think that your third question had to do with testing whether drugs that succeed in preclinical models in animals will also work well in humans. And as you heard last night, you know, we've cured a lot of mice with cancer, and those drugs, many of them have done nothing for human cancer. And that's why we have set up, along with many other people, other centers, ways to look at human cells or to recreate what we see inside a patient. So we take out a tumor, and we also take out the tumor microenvironment along with them. So the T cells and the innate cells and the adaptive cells, to try to replicate what it really looks like inside the patient. And we test our drugs on those organoids, is what they're called, organoids. And I think it's going to be helpful in translating what we see in preclinical animal models to human beings, because there's a lot of difference between the two of us.
Io avrei una domanda.
I have a question.
I have a question.
Okay. There's one over there too.
Qual è stato come si può quantificare l'impatto?
How could we quantify the impact of the recent epidemics? How can we quantify, and what was the impact of the recent epidemic that we experienced in terms of prevention and treatment of cancer? So what did we learn from the recent pandemics in the field of cancer treatment and prevention?
So the question is, has the recent pandemic, COVID-19, taught us something about how we approach cancer? And it's a really good question, because I think we all have learned a lot from COVID-19. And one of those lessons, which is good for our patients, is the onset of Zoom. Now, we all get a little tired of Zoom, but for patients who can communicate that way, it is efficient for them. They don't necessarily have to come in, see their doctor, then drive back home. They can just do it by telehealth. That's not always possible. Patients come in, they need infusions of drugs and so forth. But I think the other thing it's told us is that we can succeed. The COVID-19 vaccine was made in 10 months. I like to quote something that was written in the Boston Globe, and it said, you know, the West Coast is really good at making social apps, but the East Coast developed the COVID-19 vaccines, which have saved so many lives. Boston is an incredible place to be because it is a huge life scientist ecosystem. There are many academic medical centers and hospitals surrounded by biotech companies and by pharmaceutical companies. And so it's really a place where research is cherished and upheld and supported. And it makes it easier to go from fundamental biological research and translate that fundamental research into a new therapy for a patient.
Is there any other question? Just one here and then another there, please. Good evening. We know that the current treatments against cancer also have side effects. So my question is, considering these combination therapies that you mentioned, are we running the risk of increasing the number of side effects, given that there are many treatments used, or do the side effects that stem from these combination therapies remain the same number, let's say, do not increase?
So this is a really wonderful question, and one which we all have to think about. And the question is, what happens if you combine all these different therapies, each of which does have some toxic side effects? Are we going to double or triple or quadruple the toxicity that these patients will see? It's a really good question. And, you know, I would say we really don't know the answer yet. What we have been able to do is to understand what the toxicities are and how to treat them. So, for example, in immunotherapy, there were a certain number of patients whose T cells became overactivated. And that led to basically an autoimmune-like disease where you're attacking your own body. And that was because those T cells were pouring out inflammatory cytokines. These are small molecules that are critical. So patients were developing skin rashes, colitis, in the worst case, myocarditis, and thyroiditis. Now we figured out how to calm that down, which is to give them blocking antibodies against those inflammatory cytokines. So we've largely solved that problem. There are very few toxicities for genomic mutations, for drugs that target a genetic mutation. Those are really pretty safe. There's an occasional bad effect, but mostly they're not toxic. And of course, for CAR T cells, that's an exaggeration of immunotherapy because you're putting in potent, extremely potent T cells. And that has, again, caused the death of some of the first patients who were transfused with CAR T cells. But again, that has been subdued by blocking those inflammatory responses. So, you know, every drug that we ever take is going to have some side effects. You know, it's a risk versus benefit equation. Is it worth the risk to treat a patient with X, Y, or Z? And that's a decision that you make together with the patient. But I do think a lot of progress has been made in really being able to treat those side effects. Okay.
Bene.
Fine. We can thank Professor Glimcher and close our event. We do hope that Professor Glimcher can come back and tell us what were the further improvements.
In this therapy, in these various therapies, and we were really happy to have you here, and we thank you again for your wonderful presentation.